Hello students, today we will discuss about the changes in the second week of development. When you will reach on the 7th day of fertilization, you will realize that the process of implantation has already been started in the form of partial attachment. Now, once there is a partial attachment of the blastocyst happened, now it will start to penetrate deep and deep inside the endometrial wall. So, there are changes starting into the outer layer, that is in the trophoblast, and simultaneously the changes also starts into the inner cell mass, that is in the embryoblast. So, first we will discuss the changes in the outer cell layer in the second week of development. So, my dear students, the question comes is formation of primary villi. So, whenever you are talking about the formation of primary villi, you have to keep this thing in mind that the primary villi form on the second week of development. So, how the primary villi form? So, my dear students, what is happening that this is your blastocyst. Now, this blastocyst is surrounded by a layer is known as trophoblast, and this trophoblast divide into the two. Now, on the inner side of the trophoblast, where you are having the inner cell mass, now these trophoblasts are known as polar trophoblasts. So, we know that implantation starts from the polar end or embryonic pole. So, what will happen that first these trophoblasts will differentiate into the two layers. Now, one layer which is formed inside is known as cytotrophoblast, and there is a formation of another syncytial layer is syncytiotrophoblast. So, first step is differentiation of trophoblast. Now, for the formation of primary villi, first this syncytial layer has to increase in the size. So, syncytial layer will release the enzymes. These enzymes will damage the decidua and more invasion will take place. So, with the time this syncytial layer will increase in the size and it encloses the whole developing blastocyst. Clear? So, now there is a formation of syncytial layer all around this developing blastocyst. Now, what will happen? First step is differentiation of the trophoblast. Then, now there is a formation of syncytial layer completely around your blastocyst. Now, there is a appearance of lacunae. So, these lacunae first appears near the polar end. Now, what will happen here that in this area you will have the appearance of the spaces. Now, these spaces are smaller in size initially, and later on the spaces will join to each other and they will form the large spaces. Now, my dear students, these large spaces are known as lacunae, and this stage of blastocyst is known as lacunar stage. Now, what will happen next? That there is a formation of trabeculae. Now, what is trabeculae? Now, see here what you are able to understand that this is one lacunae, this is another lacunae, and between the two lacunae you are having this cord of syncytial layer. Now, just try to assume that you are in a very big building, or in that building you are having a hall with the multiple pillars. Now, you can rotate around the pillar, you can rotate around the another pillar. So, these two pillars which are placed here, they are the pillars of made up of your syncytiotrophoblast, and the space between the two pillars is known as your lacunae. So, these pillars, which is the area between the two adjacent lacunae, is made up of syncytial layer, and these areas are known as trabeculae. So, what is trabeculae? Trabeculae is a area between the two adjacent lacunae, and trabeculae are made up of syncytial layer. Now, what will happen? The core of trabeculae pierced by the cytotrophoblast. So, these cytotrophoblastic cells start to pierce, and these cells will migrate inside the center of this trabeculae. So, now the pillar which is initially purely made up of syncytial layer, now in the center having one more layer, and that is your cytotrophoblast. So, now this structure, if you will cut, how it appears? You will have in the center a cell of cytotrophoblast, and this cytotrophoblast surrounded by a layer of syncytial trophoblast, and here is the one lacunae, this is the another lacunae. So now this structure, which is centrally having the cytotrophoblast surrounded by syncytial layer, is known as primary villi. Clear? So what is the steps which are taking place in the primary villi formation in second week? First, the trophoblast differentiate into the syncytial layer and cytotrophoblast. The syncytial layer will have the lacunae, which is known as lacunar stage. Then area between the two adjacent lacunae is purely formed by the syncytial layer, which is known as trabeculae. Now in the trabeculae, the center part is pierced by the cytotrophoblastic cells, and now this structure, which is having core of cytotrophoblast surrounded by syncytial layer, is known as primary villi. Clear? So this is the most important change which will take place in the syncytial layer. Clear? Now the another change which will take place here is formation of BiLaminar germ disc. Now these are the changes which are taking place inside the developing blastocyst, and you know that in the developing blastocyst you are having a group of inner cell mass. Now this is the group of inner cell mass which lies at the one end of this blastocyst. Now this inner cell mass or embryoblast cells will rearrange themselves, and when they will rearrange, first there is a formation of a flat cell layer, and that flat cell layer is going to form your hypoblast. Clear? So what will happen? This is your blastocele. So these cells will rearrange, and here you will have a flat cell, or you can say low cuboidal cell layer. Now this cell layer, which is facing this blastocele cavity, is known as hypoblast. So if somebody will ask you which layer form first, hypoblast or epiblast, answer is hypoblast. So the inner cell mass first will form a thin flat cell layer, is known as hypoblast. Now what will happen, that the other cells of the embryoblast become columnar. So these remaining cells which are here, they will rearrange themselves and they are become long in size, and these long columnar cells are known as epiblast. Clear? So this is the epiblast layer, and the lower one is known as hypoblast layer. Clear? So now this is the formation of BiLaminar germ disc. Now simultaneously there is a formation of amniotic cavity and primary yolk sac. So what will happen? The epiblast cells got a small cavity in between them. Means when you will see this epiblast layer, now between the epiblast layers there is a appearance of a small cavity here. Clear? So what is the first step? The embryoblast cells rearrange themselves. First they will form a flat cell layer, is known as hypoblast. Then they will form a columnar cell layers, is known as epiblast. In between these columnar cells there is a appearance of the cavity. Now gradually the cavity will enlarge, and once the cavity will enlarge, there is a splitting of the epiblast cells. One cell will remain in the floor and one cell will go into the roof of this cavity. So the cells will split and they will then surround the cavity. So what is happening now? The floor of the cavity is lined by your epiblast cells, but the roof of the cavity is lined by the split cells of the epiblast, which are now known as amnioblast cells. Clear? So what happened here? Now if we will draw that diagram again, so this is your blastocyst. Now blastocyst is lined by the cytotrophoblast cells. Now here you will have the flat cell layer. Now this flat cell layer is your hypoblast. Now here you will have the long columnar cells which are epiblast, and there was one more layer which has already been split. So if we will draw that split cells, so these split cells will now come here and they are known as now amnioblast. They are known as amnioblast. So this cavity is known as amniotic cavity. So now my dear students, when you are reading the formation of bilaminar germ disc. Simultaneously with the formation of bilaminar germ disc, on the one side there is a formation of long columnar cells, that is epiblast, and along the epiblast there is a splitting of the cells and a cavity will appear is amniotic cavity. So this is a question of your exam. Amnioblast cells arises from, answer is by splitting of epiblast cells. Some book says that these amnioblast cells comes by splitting of cytotrophoblast layer. But my dear students, the most of the books suggested that it is not from the cytotrophoblast, it is actually by splitting of the epiblast layer itself. So this is my suggestion, that if you are having the question, the origin about amnioblast, you should always speak epiblast rather than cytotrophoblast. So now this is known as amniotic cavity. Now the hypoblast cells also produces a new generation of cells. So now we'll move to the changes in hypoblast. Now this hypoblast layer now will produce a cell, and these cells will migrate towards the blastocele cavity on inner side of this cytotrophoblastic layer. So these cells will form a new layer, and this new layer lies inside your cytotrophoblast. Now this new layer is known as Husser's membrane. What is that known as? It is known as Husser's membrane. So what is Husser's membrane? Husser's membrane is nothing but it is a cell layer which is formed by migration of the cells of hypoblast, and these cells lies on inner side of cytotrophoblast. So my dear students, now this blastocele cavity, which earlier lined by the cytotrophoblast cells, is no more known as blastocele, because it is now one-sided lined by the Husser's membrane, and on one side it is lined by the hypoblast cell layer, and now it is known as primary yolk sac. This primary yolk sac cavity is also known as primary umbilical vesicle. Clear? So whenever you are writing about the bilaminar germ disc, you have to understand that first there is a rearrangement occurs in the inner cell mass or the embryoblast cells. First there is a formation of hypoblast, which is a flat cell layer. Then the remaining cells will become columnar in size, which are known as epiblast cells. Then there is a appearance of a cavity inside the epiblast cells. These cells will split, and the splitting will completely circle or completely line the cavity. The roof is now formed by the epiblast cells, which has been changed into the amnioblast. The floor is still lined by the long columnar epiblast cells. Then there is a changes occurs in the hypoblast cells. Hypoblast cells also will produce a new cells, and these cells will migrate and form a layer inside the cytotrophoblast, and this cavity is now no more known as blastocele. Now it is named as primary yolk sac. So when the bilaminar germ disc will appear, on one side it is having a amniotic cavity, and on other side it is having a primary yolk sac. Now what will happen in the next step of the second week of development? There is a formation of extraembryonic mesoderm. Now this is known as extra because this mesoderm is not going to form any product which persists in an adult body. Whatever the products arises from the extraembryonic mesoderm, they are helpful in the nutrition of the developing embryo, and they will not give rise to any body organ. Clear? So they are extraembryonic. If we are having any body organ arising from the mesoderm, then it has known as intraembryonic mesoderm. So this is the extraembryonic mesoderm, and the first question which you have to understand, that where extraembryonic mesoderm lies. So my dear students, I just told you that there is a formation of primary yolk sac, and this yolk sac is lined by the Husser's membrane and this layer of cytotrophoblast. Now there is a gap. Now the gap is present between the Husser's membrane and cytotrophoblast, and in this gap you will have the appearance of a new layer, and this new layer is all around present between the both yolk sac, amniotic cavity and your embryonic disc. So what will happen here? The primary yolk sac wall, the wall of the primary yolk sac will secrete the cells or a new generation of the cells. And these new generation of cells occupy the area inside the cytotrophoblastic layer. So what will happen? The bilaminar germ disc with amniotic cavity in primary yolk sac surrounded by a new cell mass which is known as extraembryonic mesoderm. Clear? So if you will see here what is happening, now this is your cytotrophoblast, clear? And here this is your bilaminar germ disc. On one side you will have the amniotic cavity, on other side it is Husser's membrane, and this is your primary yolk sac. Now in this region you are having this extraembryonic mesoderm, and because of this extraembryonic mesoderm formation, what you are able to understand now, the cytotrophoblast completely split from this yolk sac, amniotic cavity and germ disc. Clear? So this is the area which has been occupied by the cells of extraembryonic mesoderm. And what is the source of the cells of extraembryonic mesoderm? It is again from the wall of yolk sac, and the cells in the wall of yolk sac are coming through the hypoblast. So there are again a second thought also. Some book says that this extraembryonic mesoderm arises from this outer cytotrophoblastic layer. Clear? But again, I will suggest that it is not like that. It is actually the hypoblast cells which are continuously migrating, and along with that the yolk sac cells are going outside and they will surround this whole developing disc. Clear? So first the primary yolk sac wall secretes the cells, and these cells will come between the cytotrophoblast and amniotic cavity and yolk sac area. Now what will happen next in this extraembryonic mesoderm? Now there is a formation of cavity. Now my dear students, when you will talk about the cavity, this cavity initially form in the small groups or in the form of lacunae, and then these lacunae will fuse with each other and they will create a big cavity. But this cavity is not present all around. It is absent at one part. So how to draw this cavity? Now this cavity is formed in such a way. Now when you are drawing this cavity, you will realize that this cavity splits the extraembryonic mesoderm into two layers. So this is your cavity. Now here you can see that this cavity has divided this mesoderm into the two layers. Now where is the two layer? Now let's see the two layer. This is the one layer which is present on the inner side of cytotrophoblast. This is the one layer which is lining the yolk sac, and this is the one layer which is lining the amniotic cavity. So the cavity splits the extraembryonic mesoderm into the two layers. What these layers are known as? One is known as splanchnopluric extraembryonic mesoderm, and one is known as somatopleuric extraembryonic mesoderm. So what is splanchnopluric? The layer of extraembryonic mesoderm which present around the primary yolk sac. That means this is your primary yolk sac, and around the primary yolk sac, this layer of extraembryonic mesoderm is known as splanchnopluric extraembryonic mesoderm. While the mesoderm which is present around the amniotic cavity and which is present just inside the cytotrophoblastic layer is known as somatopleuric extraembryonic mesoderm. Clear? So what happened that this cavity is appearing in the extraembryonic mesoderm, and this is dividing into the two parts. One is somatopleuric and one is splanchnopluric. Now there is a one more term comes is chorion. Now what is chorion? Now chorion means the cytotrophoblastic cell layer with somatopleuric extraembryonic mesoderm. So this is your cytotrophoblastic cell layer and this is your extraembryonic mesoderm. So now this thickness is known as chorion and that's why this cavity is known as chorionic cavity. What is that? Chorionic cavity. So when you are talking about the changes in the second week, first there is a formation of the amniotic cavity and yolk sac. Then there is a appearance of extraembryonic mesoderm outside the cavities. Now in the extraembryonic mesoderm there is a appearance of another cavity. Now this cavity is going to divide the extraembryonic mesoderm in an inner lining and outer lining. The outer lining is known as somatopleuric. The inner lining is having the two part. One is outside the amniotic cavity is also known as somatopleuric and the lining outside the yolk sac is known as splanchnopluric. Clear? But my dear students, when you are forming this cavity, you can see that at one end this cavity is absent. Now here what is happening that this is your cytotrophoblastic layer, this is the germ disc and these are the cells of extraembryonic mesoderm. So this layer of extraembryonic mesoderm which is connecting the developing germ disc with the outer cytotrophoblastic shell is known as connecting stalk. What is that? It is known as connecting stalk. Now this connecting stalk is important structure because in this connecting stalk later on the arteries will develop, the vessels will develop and it will give nutrition to the developing germ disc. And this is a future area of umbilical cord. Clear? So this is the important change which will take place in the development of second week. Now in the second week there is a one more change will take place, that the primary yolk sac convert into the secondary yolk sac. So the next change is changes in the primary yolk sac. So the hypoblast cell layer now produces one more new cell layer. What is happening here? Now this is your primary yolk sac, this is your hypoblast. Now this hypoblast cell will produce one more new generation of cell. Now these new generation of cell will go and they will line a very smaller cavity as compared to the yolk sac. So here what you are able to see, first there was a blastocyst lined by the trophoblastic cells. Later on there is a formation of Husser's membrane. Now the cavity name has been changed to the primary yolk sac. Now there is a formation of one more new layer by the hypoblast cells and now a cavity become very smaller in size and this is known as secondary yolk sac. So my dear students, what will happen with the primary yolk sac? Primary yolk sac will divide into the two cavities. One is the smaller one. Now this smaller one which is adjacent to the hypoblast is known as secondary yolk sac. But the majority portion of this primary yolk sac pinched off, and this majority portion or the larger portion of the primary yolk sac which has been pinched off is known as exocoelomic cyst. What is that? Exocoelomic cyst. And this cavity which is now known as chorionic cavity will enlarge in size. And when this cavity will enlarge, it will become like this. This cavity will enlarge in such a way, and once this cavity will enlarge, what will happen? This cyst will float into the chorionic cavity. So sometimes you have this question that where will you find the exocoelomic cyst? So exocoelomic cyst will float into the chorionic cavity. So first the hypoblast cell layer will produce a new layer of the cells, and these new cells lies inside the Husser's membrane and they will create a very smaller cavity which is known as secondary yolk sac. So the primary yolk sac also having the two part. One I told you, a smaller one is the secondary yolk sac, and the larger portion or of the residual part of the primary yolk sac which has been pinched off is known as exocoelomic cyst, and this cyst is present into the chorionic cavity. Clear? Now there is one more change will take place, formation of precaudal plate. So when you will see the development of the germ disc, what is happening that this is your germ disc, which is one side is having amniotic cavity and another side you will have the secondary yolk sac now. Now here you will have the long columnar cells which are of epiblast, and here you will have the flat cells of hypoblast. Now what will happen at one end of this hypoblast layer, the cells will change their nature and they also become longer in size, and this cell area is known as precaudal or procaudal plate. So the first question comes is that precaudal or the procaudal plate arises in which layer of germ disc? Answer is hypoblast cell layer. So there is a differentiation occurs in the hypoblast cell layer near one end of the disc. Now this end where the hypoblast cells modify in the long columnar is going to form your future mouth. So appearance of precaudal plate decides the cranial end of the plate. So now in nutshell, what are the changes will take place in the second week? The first change is that I told you, because we are talking about the second, so you will have the word 2 in the second week, and that's why it is known as week of 2. So there is a formation of 2 layers from trophoblast, cyto and syncytio. There is a formation of 2 layers from the embryoblast, hypoblast and epiblast. There is a formation of 2 layers of extraembryonic mesoderm, somatopleuric and splanchnopluric. There is a 2 cavities will form, yolk sac and your amniotic cavity. There are 2 types of the yolk sac form, primary yolk sac and secondary yolk sac, and primary yolk sac divides into the 2 types of the cavity, secondary yolk sac and exocoelomic cyst. Clear? So my dear students, these are the some important changes which are taking place into the second week of development. So now at the end of this class, you have the brief idea about the second week of development, that once there is a fertilization will take place. After the 7th day of fertilization, the implantation will complete. It will change from partial to the deep implantation. There is a formation of the primary villi. Then simultaneously in the embryoblast, the changes will take place. There is a formation of BiLaminar germ disc with the formation of amniotic cavity and yolk sac, and there outside the changes will take place in the form of appearance of extraembryonic mesoderm, which will later on divide into the 2 layer by the appearance of the cavity. So this is all for the session. Thank you.